Good morning, dear students. In today's lecture, we will talk about different structures of proteins. We have talked about amino acids, and I have given you a general introduction of proteins also. Proteins are the biopolymers of alpha amino acids. Only alpha amino acids play a role in synthesis of protein. Term protein or name protein is derived from Latin language. It's a Latin word proteus, which means of prime importance. So proteins are the biomolecules having prime importance. They are very highly essential for life. More than 80,000 types of proteins are found in living beings. And for that, only 20 alpha amino acids are utilized. So their sequence in a proper manner will decide the type of protein formed. So starting with primary structure. What is primary structure? The sequence of alpha amino acids will decide the type of protein formed. So primary structure is nothing. It is simply the arrangement or the sequence of alpha amino acids that is going to decide which type of protein will be formed. If we change even a single alpha amino acid, then it will form defective protein. The protein which is having no utility in our body. So sequence is absolutely important. So what is primary structure? Primary structure is the sequence in which alpha amino acids are arranged in a protein. This is called the primary structure, and the amino acid sequence of protein determines its function and is important for its biological activity. So sequence is absolutely important. Everything is decided by the sequence of alpha amino acids. Then comes the secondary structure. In primary structure, we are having linear arrangement of alpha amino acids. Amide linkage is there between the different alpha amino acids. C double bond to NH. Unit is there. So condition required for hydrogen bonding is there. So when a single chain of alpha amino acids undergo hydrogen bonding, it is your intramolecular H bonding, then coiling is developed. So here, due to development of H bonding, coiling is there. Earlier in primary it was a linear chain, straight structure. Now force of attraction is developed due to H bonding, so coiling is there. So this structure is called alpha helix, present in keratin. It's a dead protein. Now, when more than one polypeptide chains are involved in protein formation, then between the different polypeptide chain, H bonding can be developed. This H bonding is called intermolecular H bonding. So when intermolecular H bonding is there between different polypeptide chain, then we obtain the second type of structure. In secondary structure is called beta pleated sheet structure. Beta pleated sheet structure, and is present in silk. Silk is a type of protein. So remember, first one is simply the sequence. Second one is due to H bonding, coiling is there. A force of attraction is there. Till secondary structure, the proteins are non-functional. They cannot perform any function in our body, or we can say they don't have any biological activity. But when the secondary structure gets converted to tertiary structure, then the protein acquires activity. It becomes functional in our body. And how tertiary structure are obtained? Further coiling in secondary structure converts the secondary structure into tertiary structure. More coiling is there. Different forces are there which are responsible for this coiling. And the forces are H1, it was there, disulfide linkage, van der Waals force of attraction, and electrostatic forces of attraction. When further coiling is there, to provide stability to the coil structure, different forces comes into existence. And these forces are H bonding, disulfide linkage, van der Waals force of attraction, and electrostatic forces of attraction. Here, the protein has attained functional activity. All the enzymes have tertiary structure. Then comes your quaternary structure. What is quaternary structure? It's very simple. When more than one polypeptide chain are there, then their arrangement, or we can say space arrangement with respect to each other, is called the quaternary structure. So here in quaternary structure we can say quaternary structure composed of two or more polypeptide chains. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. Like here, we are having three subunits. There are three polypeptide chain. Here it's the first one shown by black ink, black marker. Next one is your red one, and third one is your blue one. So there are three subunits or three polypeptide chain. So their arrangement with respect to each other is called the quaternary structure. So that's all in the structure of protein. In our next lecture, we will talk about nucleic acids. Thank you and have a good day.